nice.
Well, there is advantage to getting out because you have a.
Okay, should we get started?
Sure.
Thanks for being here. I really appreciate it. I really appreciate the invitation. I'm Betsy Quinlan. I am a neuroscientist that studies the way the brain changes with age because it's just an interesting biological question, but also with this intent to identify ways to make the adult brain act more like the brain of a juvenile. Okay, so I talk about my work as we are working to identify ways to rejuvenate the human brain. I was born in the Midwest. I did my undergrad at the University of Iowa. I have a bachelor's in science and biology from the University of Iowa. I have a PhD in neuroscience from the University of Illinois, Chicago. And then I left the Midwest in 1993. We, faculty academic scientists like myself, do postgraduate training before we have the chops to have our own labs. So I did some additional training at the University of Virginia, and then some additional training at Brown University. And then I was a faculty member at the University of Maryland for 22 years. 2 years ago, I moved back to the Midwest to join the faculty of the University of Wisconsin-Madison, and I'm now the chair of the Department of Neuroscience. And so I am thrilled to be back in the Midwest. I love the Midwest. I love Madison. I love the university, and I love my job. So it's just a great move all around.
But do you love winter?
You know, I'm not afraid of winter. I did grow up here. I decided this year, I mean, we've had 2 really mild winters, and I thought, oh, these Wisconsin winters that everybody's been warning me against, that's just so that everybody doesn't move here. You know? But I think we're going to have a real winter this year. Welcome. So the title of my talk is this very provocative, Can We Rejuvenate the Aging Brain? And I would not have titled my talk this way if I didn't believe that the answer was yes. So let's just talk, and I want this to be a dialogue, so please interrupt me, ask questions, ask for clarification, and if you want more details, please ask. If you want less details, that's okay too. So let's just think about what we mean by aging. I think we all have an intuitive sense of what it means to age. Okay, there are changes in our physical abilities, including strength, mobility, our ability to heal, changes in our skin and our bone health and density. There are also mental and emotional changes. Okay, these are the ones that I'm focused on as a neuroscientist. Cognitive vitality, this is a very carefully chosen term. It's not cognitive, it's not memory, it's not the ability to make memories per se, it's vitality. Being flexible, adaptable, resilience and positivity, purpose and enjoyment, honesty with needs, and then very importantly, social and lifestyle. And so there are changes in our relationships. We tend to, perhaps not this audience, but we tend to be less involved with the community, and we tend to be less proactive about our health. Okay, so this is just this long laundry list of things that happen as we age. Okay? And there are people whose aging seems to be more accelerated relative to others. Please.
Do you have handouts or should we be taking notes?
I do not have handouts, but if you would like, I will share my PowerPoint with you afterwards. I'd be happy to send it to Ann to print out or email it to you. Okay, and that way you could just relax. All right, so we all know that different people seem to age at different rates. Right? And there's also this phenomenon of superagers. There is a well-known and very heavily studied cohort of individuals that seem to defy the typical aging process. They maintained incredible physical health and incredible mental and emotional health. And so we're going to talk a little bit about what makes a superager, and to see if there's some lessons we can learn there. I'm going to introduce you to some really basic neuroanatomy. So this is the building blocks of the brain. Okay, these are all cartoons. On the left, of course, is the way that the human brain sits in the human skull. Now, if we were to put this brain under a microscope and zoom in about 40-fold, do about a 40-fold magnification, we could see the individual building blocks of the brain. This is one of the building blocks. It's the primary building block. And this is neurons. So neurons are the primary cell type in the brain. And then if we zoomed in again another 10-fold, we could see a structure called a synapse. Synapse is from the German that means to connect. Synapsen is to connect. And so a synapse is the connection between two neurons. It's how two neurons talk to each other. And synapses, which I'm going to talk a lot about, so I want to make sure that we're all really clear on the concept of this synapse, this connection between two neurons, has a particular structure. And the structure is shown in the cartoon on the right. Okay? There's a top and a bottom. There's a part of the synapse that transmits the signal, and there's a part that receives the signal. And that's not important. Those details aren't important. It's where you have communication between neurons. And it has a particular structure, meaning we can find it in microscopy. Okay? So anything that we can find and identify over and over again is much easier to study, right? So the synapse has a particular anatomical correlate. We can find it, we can measure them, we can count them. Okay? We have something that we have a metric that we can use. On the right is a piece of a neuron. This is a dendrite. This is not a particularly important term, but in green is a piece of a neuron, and the synapses, the postsynaptic contribution of the synapses are stained yellow, okay, and those are called spines. So the only two things you really have to remember is synapse is the connection between neurons, and spine is the anatomical correlate of that synapse. We can count spines. You can see how easy it would be to count spines. On the right side there, in yellow, are spines on a dendrite of a neuron of a post-mortem subject. We can't do most of this in live. In live, we can't do this. We can't do this anatomy, this level of anatomy in live subjects. And you can count them. You can count the number of spines. We can start making predictions about how spine number or synapse number impacts cognition. Please.
You said you can't do this on people.
We could do, so I'm going to show, the next one is going to be, we can't do this level of analysis. So this is about 400 times magnified. Okay? And so you need to put the brain under a microscope. So this is a post-mortem tissue. Okay? So to do the really reduced counting of individual synapses, it has to be post-mortem. But now we can zoom back out because we can see the reflection of changes in spine numbers in MRIs. Okay? So first I just want to give you some numbers. This is just, again, connection between neurons or synapses, and many synapses occur on these spines that we can count on the right side. And just to give you a sense of our, just to give you some numbers, our brains, human brains, have 100 billion neurons. Every neuron makes about 1,000 synapses. And that means we have about 100 trillion synapses in our brain. Okay, 100 trillion.
And what amount of time does that happen?
Is a great question because it brings up my next slide. Perfect. Thank you for that. So spine number, the number of spines or the number of synapses, we're going to use them interchangeably, changes over time. And on the horizontal axis, we're going to go from pre-birth to adulthood to middle age to older age. And let's look at the data in black. So black is normal, neurotypical, disease-free synaptic density. Okay? We all start with very low synaptic density. Synaptic density peaks in early childhood. Okay? We actually make more synapses than we need in early childhood. And then between early childhood to adulthood, this light green section where there's a steep decline in synapse number, that's when the brain circuitry is refining. That's when skill mastery is developed, right? So we all know that most violin virtuosos pick the violin up as children, right? So there's something about developing expertise. Children can develop expertise in a way that's very different than adults. And part of the reason is because there's so many extra synapses, and then the synapses that you need, perhaps, to run your hand up the neck of the violin, you keep those, and you basically get rid of the ones that you don't need. Okay, so here's a normal, this is a normal adult synapse number. But there's also a decrease in synapse number towards the later years of life. So we're gonna focus on that decrease in synapse number in the later years of life. I also want to draw your attention to some of the other data on this chart. So Alzheimer's disease, which is in blue, is characterized by this acceleration of synapse loss in later life. Compare the blue to the black, okay? Incredible acceleration of synapse loss. And we know what that does to cognitive function. Schizophrenia is a disease that typically emerges in late adolescence. And this is one of the reasons why. Look at what is happening to the time course of synapse elimination between schizophrenics in green and normals in black. Okay, so there's an accelerated loss of synapses in schizophrenia. But we also understand that it's a balance. There's the right amount of synapses. Too few is Alzheimer's. Too many is also a disadvantage. And if you look at the final data point here, which is in pink, you can see that autism spectrum disorder is characterized by an abundance of synapses that are not properly tuned. So autistic people actually have more synapses than normal. And so synapse number, there's a sweet spot for synapse number. Okay, so these synapses have structure. We can count them. To go back to the human question, we can see the consequence of synapse loss because synapses have volume. We can see the consequence of synapse loss in MRI. So here's a series of MRIs that range from adolescents to young adults to middle adults to older adults. This is a side view. The first column is a side view, obviously. And the middle column is a coronal section, what's called a coronal section. So if you slice the brain like a loaf of bread, starting from the front and going to the back, this would be a slice of about the ears, right? An MR can do that. MR can take these, can take individual slices. And look at the way the brain is sitting inside the skull. This is perhaps the more obvious in the example I'm showing on the right. On the left, on A, is a young adult, and in B is an older adult. And there is the brain has shrunk, and there's quite a bit more space between the brain and the skull. Okay? The brain shrinks as we age. This is the consequence of this synapse loss. Okay? So these synapses have volume, and when you lose synapses, we can see the loss of volume in MRI. Okay? So that's how we study this in humans. We look at volume. And so there's a couple different ways you could quantify this. You could quantify brain volume, which is going to go down over time, or you can quantify the volume of space, okay, which is going to go up over time. And if we look at the volume of space, which goes up over time, let's look on the right. Typical adults ranging from 75 to 90 years old have an increase in the volume of the space. But superagers have decelerated that loss. Okay? And so the superagers are actually losing less synapses. They're losing less spines, and you can see that at the level of MR. So what can we do? What can we do to maintain brain volume and maintain synapse volume? And so to answer that question, I'm going to tell you a story. All science is based on foundational work that tells a story. And this is some work that was done by Marion Diamond. Marion Diamond is a renowned neuroscientist who was at the University of California, Berkeley, and asked a really interesting question, a profound question that has impacted the way that we think about the brain. So Marion worked with mice, as many of us do. And at the time, all mice were raised in what's called, we now call this a standard environment or an impoverished environment. Okay, it's very much like a kennel, just the basics. Okay, there's food, there's water, and there's a little nest for them to sleep in, but nothing else, just the basics. And Marion tested the hypothesis, how does the complexity or the richness of the environment impact the way the mice behave? And so she was the first person to move away from the standard environment and give the mice an enriched environment. So this is the standard now. Okay, we do not raise our mice in our experimental mice in standard environments. We raise them in enriched environments. Okay, they are full of toys that make noise and have color and move. They are raised in a group, so there are social interactions. So there's all of this stimulus for all of the senses that we give our mice. And importantly, to do this right, to do the enriched environment correctly, you change it up frequently, right? So you rotate the toys, right? You change the configuration of the habit trail that they're running on, okay? You know that this is now incorporated into the way we change toys with children, the way we reorganize exhibits at the zoo, always moving, always changing things up and always moving things around. So Marion raised half the mice in this standard environment, half the mice in the enriched environment, and then looked to see what it did to brain volume. Okay? Some of these results are on the right. Animals that are raised in the enriched environment are shown in purple, and the impoverished environment are shown in gold. And you can see that the thickness of the cortex, the brain thickness, the brain size, the brain volume is significantly different across a number of regions. The regions aren't that important, but they are regions that control motor activity, and regions that control sensory activity, and regions that control social interactions. And so I don't like the analogy of thinking about the brain like it's a muscle, but it's basically if you exercise all the different parts of your brain like you do in this rich environment, your brain actually gets bigger. I think it's more correct to say your brain doesn't shrink because it's not impoverished. There's a lot of quantification. Hundreds of labs have replicated this. This is a foundational observation in neuroscience. Hundreds of labs have replicated this. Here's a picture. This is not a cartoon. This is actually a neuron that was filled with the dark substance so that it could be completely traced in a mouse raised in a standard condition and in an enriched condition. Look how much complex just the individual neurons are. And now we're going to count the number of spines on neurons. In animals that were in the standard environment, these are the lower curves with the open circles, versus the enriched environment, which is the dark circles in the upper curves. Okay, so mice that were raised in an enriched environment have more spines, have more synapses, have more complex neuronal arbors, we call them. But are they smarter? I mean, what do we do with these synapses? Are these synapses important? Do they correlate to cognition? And so for that, we're going to move to the work of another renowned neuroscientist, Carol Barnes, who is currently the Regents Professor in the Department of Psychology at the University of Arizona. See if this is going to work. Carol designed a test 24 mice to test their memory that is now used in laboratories all over the world. Her name is Carol Barnes. We call it the Barnes maze. And so I'm going to show you a video of a mouse that is performing a Barnes maze for the first time. Look at the structure of the maze. So there's a surface that the mouse can explore. They're very curious. They explore every nook and cranny of every novel surface. But they don't particularly like being in the light. They are nocturnal and they feel safer in the dark. So they're looking for a place to hide. And so the place to hide is here in the bottom, the open port. And it will always be there. The open port will always be in the same location. So you can see that there are simple shapes taped to the wall of the room that holds the maze, and the animal learns where the escape port is relative to these signs. Okay, so builds a map and then remembers. So it's kind of fun. If I can get, let's see. See if it'll play from here. All right, we will skip that. Sorry about that. I will tell you, these have become very sophisticated. So you place the animal in a chamber on the ground with gloved hands because you don't want the chemical identity of the individual, which might vary from day to day, to differ or impact the animal's behavior. So you do this very sterile introduction of the animal to this little chamber. And it's an elevator. So the chamber brings the animal up. This is the elevator here. This is the elevator after it's risen to the top. The elevator brings the animal up to the top, and then they're faced with this, very large, relative to their body size, very large surface. They explore it, and then they accidentally run into the hiding spot. So the first encounter, it's an accident, right? But they remember it. So what's shown on the left is the amount of time is on the vertical axis, the amount of time it takes for the animal to find the hiding spot over trials. And all of the cohorts in this experiment, they all get better at it. So they all have some learning. But the differences in the groups are very interesting. So in this lime green are animals that are 5 months old. This is a juvenile mouse that are 5 months old, but they were raised in a standard condition. Okay? They're young, they learn fast, especially compared to what's in blue. In blue is a 24-month-old mouse, a 2-year mouse, raised in a standard condition. Look at the difference in the learning rates between a 5-month-old and a 24-month-old. But a 24-month-old that was raised in an enriched environment, which is in red, performs more like a 5-month-old. Okay? So this shows us that all of these things are correlated. The enriched environment and the spine density and the performance. Okay? It significantly improves learning performance. So we know it's intuitive that juveniles learn differently than adults. And one of the most dramatic examples of that is shown here in this very famous piece of work done by Horshorn et al. In 2018. Famous because it's looking at the impact of age at time of second language acquisition in a cohort of 669,000 subjects. Okay, so this is an extraordinary sample from all over the world. And so here what's on the horizontal axis is the age at first exposure to a second language, right? If you've ever seen the ease with which a child can acquire a second language and then struggled with it yourself, you know there's something very different between the way children and adults learn. Okay? And this is borne out in these statistics. Here's the fluency of an individual who's monolingual. And then here's the fluency in the second language of individuals that are immersion learners, meaning that they're learning the second language of their host country, total immersion. And to reach an accuracy that's similar to monolingual, you have to be exposed to the second language before 10 years of age, right? And fluency and accuracy goes down with age. And that's because, as I've said, juveniles and adults learn differently. With juveniles, most of their learning occurs in the input that's coming into the brain, right? The sensory input coming into the brain. That is the trigger. And that's very high in juveniles. That's a plasticity that's the highest in the juveniles. That's not what drives learning in adults. In adults, what's driving the plasticity is the information coming the other direction, the information that's coming from the internal source, from your brain. It's your brain predicting what's going to happen next. Okay? That is what drives the learning. Okay? So let me say that again. In young learners, the trigger for the learning is the sensory input that's coming into the brain. In adults, the trigger for learning is your brain predicting what is going to happen next. Okay? Our newest understanding of the brain is that it allows us to predict the elements of our environment, especially the elements that might be threatening. Right? So we walk through our environment saying, nothing to see here, no threats there, no threats there. Right? We evolve the ability for very good threat detection. I'm going to talk about this. If this is not clear, I'm going to talk about it a little bit more. I just want to introduce it, that there's very, very different pathways of information flow. So I'm going to ask you a question. What do you remember from yesterday? Well, if you're like me, you may remember what you eat, what you had to eat. I often do, because that's often pretty important to me. But I don't remember some of the other more mundane aspects of the day. I remember who I talked to. I always remember who I talked to, because that's important. But those are little snippets of a very long day. So I'm not remembering more than I'm remembering of a single day, right? We don't remember most things, okay? That's a good thing. Because the trick to good cognitive function and good learning is to filter out the unweighted memories, right? I'm going to talk about how to weight a memory next, okay? But most things you don't want to remember. Most things you don't want to use up storage capacity to remember some of the more mundane things of the day, right? You want to weight specific memories with some sort of tag that says, hey, this is important, remember it. You want to let the other things go. There are a very rare number of people, maybe 25 in the world currently, that have a condition called hyperthymesia. And in this case, the individual remembers every single detail of every single day, every mundane sound and sight. Right? And this is as debilitating as losing memory, right? It's a sweet spot. We do not want to remember everything. Right? So when we wish for better memory, we have to have a caveat that we don't want to remember everything. We want to remember just the things that we can tag as important to remember. We have to have some sort of way to rate our experiences to say, hey, this is important. Okay, so it's equally important to weight and filter incoming information. Remembering everything is maladaptive, and so the question now is, how do we attach a weight to a specific memory, okay, especially in adulthood? And the answer to that is to engage a fight or flight response. Okay? I'm going to explain this. I know that sounds aggressive. It's not as aggressive as it sounds. Okay? We know that when we perceive, okay, so this is important, it's the perception of threat. It's your perception of a threat. When we perceive a threat, we go into what's called fight or flight mode. Okay, that is our body preparing to survive the threat. And so pupils dilate. That helps limit your focus. You actually lose, if you're having a very strong fight or flight response, you lose peripheral vision. You get tunnel vision. It focuses your vision. Your heart pumps faster because your body's getting ready to run, to fly, or to fight. Okay? So you need to get blood out to muscles. We actually stop our digestive processes. There are many internal processes that stop because your body is shunting all its energy to the fight or flight response. You stop digesting food, shunt it all to get ready to fight or fly. You tense up your muscles in anticipation of springing up, and your respiratory rate goes up. Okay, so we're bringing blood and oxygen to our muscles. We also release a number of chemicals, adrenaline and cortisol. Okay, and adrenaline and cortisol are associated with stress response. Okay? But we know that there are many gradations of stress. So this is shown on the right, okay, where we go from zero stress, which we could also call boredom, no stress at all, to really high stress on the right, which is burnout. Okay, we know chronic high levels of stress are detrimental to mental and physical health. But there is a lot of room in between the left and the right side of this graph. And there's this sweet spot in green, happy face in green, which is called the stretch zone or the optimal zone, where you are mildly stressed. And this is very good because the mild stress has your body release adrenaline and cortisol, and that's what weights the memory. Okay? The adrenaline and cortisol weights the memory so that you store it. And so what do I mean by optimal stress? I mean, this is not stress perhaps in the most common term. It's the kind of stress like navigating in a town you've never been to before. It's annoying. It's a little disquieting. Okay, but it's not all out stressful. It's just a little uneasy. That level of uneasiness, your brain loves that level of uneasiness. Loves it. Okay, it's very good for memory. So without going into too much of the details, but I'm happy to answer questions, when you're in this mild optimal stress, your brain secretes all kinds of neurotransmitters, all kinds of chemicals, because remember, again, our brain is a predictive organ where we're walking around our routine saying, nothing new here, no threats. Nothing new here, no threats. But if you're in a novel environment, you're hyper-vigilant because you want to know if there are any threats in the environment, right? You're very tuned in. Your senses are very tuned, and you're hyper-vigilant. And these chemicals, this is a synapse, this is the spine, this is a synapse. These chemicals cause synapse production. So when these chemicals are in your brain, you make new synapses. And when you make new synapses in the presence of these chemicals, they store the experience that you're having. Okay? So think of it like a healthy kind of PTSD. I don't like the analogy, but you know, in post-traumatic stress disorder, there's a very stressful traumatic incident, and everything that happens at that moment is just ingrained in that person's memory. This is a very mild, healthy form of that. A little bit of novelty. Let's not even call it stress, let's call it novelty. A little bit of novelty helps you to remember. So, how do we get there? Right, so if this is a pathway to rejuvenate the adult brain, how do we prioritize novelty? Well, there's many ways. You've been thinking about that since I mentioned it. You can think of ways you can prioritize novelty. I mean, we were wrong. For a long time, we thought that routine was good and healthy. Okay, routine sort of lulls our brain into a stupor. Okay? Our brain does not like routine. Our brain likes novelty from the littlest things. So I'm gonna skip some of this. I'll come back if we have time. So there are many ways. We're going to talk about many everyday ways to enhance novelty in our lives. But just as an example to sort of make the point, think about how entertaining travel is and how good it makes you feel. One of the reasons that travel, near or far, someplace different, one of the reasons why traveling is so enriching is because everything in the environment is novel, right? You are flooding your brain with novel sights, sounds, smells, people, languages, everything about it is novel, right? So you're just immersing yourself in novelty. And there's quite a lot of literature on this as a way to enhance novelty. But this cartoon depicts the regions of your brain that are enhanced, that are activated while you travel. Okay? And the take-home message is every part of your brain is activated while you travel. Every part of your brain is having this experience. They're not saying, wow, this is wonderful. That's our interpretation. What they're saying is, I've never seen this before. This is new. What's that new smell? What's that new sound? The novelty. Your brain is registering it as novel. And then here's another, this is almost an advertisement about how good traveling is for your brain. Brain regions activated during travel. Well, the frontal cortex, the front part of our brain here, is involved in decision making. The hippocampal and parietal lobes, which are in the center of the brain, are involved in making maps and navigation. Our reward centers and our pleasure centers are all activated by travel. Let's think about some ways to prioritize novelty that are easier to implement. Okay. Break a routine. Even driving a different route from point A to point B is novelty. Shopping in a different grocery store. You know that little bit of unease that you get when you're in a grocery store that you're not familiar with? That is the novelty that your brain likes. Just that little bit, like you have to work at it. It's not routine. You have to work at it. So break a routine, give yourself daily challenges, explore a new environment, say yes to one small thing, start therapy with curiosity. I pulled this for a website that was maybe targeting a slightly different topic and was advocating for therapy. I'm not advocating for that. I'm saying, think about it. Break your routine. Challenge yourself to do something new. 1 5-minute challenge a day. We're talking about little steps, little improvements. Explore a new environment. Say yes to one new thing. Read a book outside your genre. I mean, I'm so guilty of this, right? I know exactly what I like. I like historical fiction, and if it's not historical fiction, I just put the book down. Right? I'm not introducing any novelty to. I'm not taking my own advice, right? Read outside your genre. Try something new, even a foreign film with a voiceover or something. There's still that disconnection. Anything that gives your brain something that's different than what it predicts is going to happen. attend workshops, take a different route, chat with a stranger. So just think about that. There are all kinds of ways to cultivate novelty. Eat something different, cook something different, try something different. So that's one, how to rejuvenate your brain. Prioritize novelty, seek it out. Another one is that your brain loves streaming. Okay, so what that means is, and it doesn't have to be walking, but imagine that you're walking. You're walking at a certain pace, and the world is streaming past you. When the world is streaming past you, it's streaming past you at a certain pace and at a certain rhythm, right? So if you imagine your brain moving through space, things are coming in at a certain pace. New trees, new house, trees, isn't it? Things are, the visual world is streaming past you at a certain rhythm. The rhythm is the rhythm that you're self-generating with your own steps. So you are setting a rhythm to the way that the visual world, doesn't have to be visual, I'm a visual scientist, so I always go to vision. It could be, all senses. Your senses are streaming past you in a rhythm that you yourself generate because you are walking. This could be on a treadmill. This could actually even be in a virtual environment. But your brain loves streaming because when your brain is streaming, all of the regions, many regions of the brain oscillate at the same frequency. So your whole brain starts humming. One of the best things to get your whole brain humming in sequence is dancing. Because dancing engages auditory, visual, motor, and there's a rhythm. So the thing that gets the largest part of your brain that is oscillating at the same frequency is dancing, walking, activity, all of these things. I mean, exercise is good for you for many reasons, but you probably hadn't thought about what the exercise was doing to your sensory perception and the correlated activity that's happening in your brain. And this, having multiple brain regions being co-active, because you're driving the rhythm with walking or dance, is very good for creativity. And so we've known since Plato that going for a walk is a good way to work through a sticky problem or come to decisions. Okay, so this type of streaming is very good for creativity in all avenues of creative process. And I have one more. Your brain loves social interactions. It doesn't have to be. There are so many types of social interactions. So even more than there used to be. Right? Social interaction could be a phone call. A social interaction could be something online, a Zoom call or a group. But of course, there's social groups and one-on-one interactions. Okay. We like to interact with other humans. Right? There's many reasons for that. To the list that you're already aware of, I'll add, maybe there's some lack of predictability. You never know what the person you're talking to is going to say next. Maybe that helps. Maybe all conversations are novel in a way. Maybe this is a way to use social interactions as a way to introduce novelty. And then this is as much for my own, I'm giving myself advice, but human interactions and social interactions are dependent on having good hearing. And many studies have shown that hearing loss and the accompanying social isolation helps to accelerate brain atrophy. And so one of the best things that we can do for ourselves is get our hearing checked and get hearing aids if necessary. That helps you to stay social and it prevents brain atrophy. So this is shown. Hearing aids prevent brain atrophy. Right? It keeps you engaged. It helps with the social isolation. So this is a summary slide. Prioritize novelty, as I've said. Prioritize walking or visual streaming. and prioritize human interactions. And these are all ways to promote synapse maintenance or synapse formation, and to maintain brain volume and cognitive and emotional function. I'm just going to take just a couple quick minutes, and then I'm happy to take questions. There are a lot of people working on the basic science of aging. This is a paper that just came out just a couple days ago. All of the cells in our body have little batteries called mitochondria. And the mitochondria also age. So all cells of our body make less energy with age. And this crazy, now we're getting into sort of a science fiction approach. This group used nanoparticles, so inert particles, to stimulate stem cells into mitochondria, all in a dish, and then have the nanoparticles deliver the new young mitochondria into the adult brains and replace the old batteries with new batteries. So this is the kind of stuff that people are working on. It is decades. It takes decades to go from this to clinical practice. Okay? And I have sort of an ulterior motive here. Of course, funding for basic science has been very much in the news. Okay? We are very much fighting for our lives. Everything I said to you was brought to you by basic science. These are individuals that are working in non-human subjects in labs, primarily with either cells or with animals, asking very fundamental questions that takes on average 15 years to become mainstream. I would say that, have you heard of baby Einstein or baby Mozart and all of these things where you could enrich your child in utero? I would say that all goes back to Marion Diamond's work 30 years ago. It took 30 years to go from the work in her lab to becoming something that we accept. I think of Marion Diamond every time I'm at the zoo, and I see all of the swings that they have in the animal enclosures, right? That's Marion Diamond too. And so just a plug for basic science. I also want to say that one of the reasons I love my job so much is that I have an opportunity to work with some fantastic people. This is a subset of my faculty that I'm showing you to show that we have experts in stem cells and Alzheimer's and movement, hearing, decision-making, cardiac function, epilepsy, vision, seizures, autism, and their ALS. They're all terrific speakers, so I hope that you reach out to me and invite one of them to come some other time to talk to you about one of these topics. And I'm going to end it there. Here's my email. Ann has it. It's pretty simple. Betsy Quinlan at Whisk. If you Google me, you will also get my address. I am. This is my first outreach talk. And so if you have feedback, I'm always open to feedback. I'm going to let you ask some questions in a second. But I also have, now that I'm in my third year as chair, I've decided that part of my mission should be to do more outreach and bring basic science to the people of Wisconsin. And so if you have connections or ideas of other groups that I can approach so that I can give a similar talk in these other venues, please let me know. I'd really appreciate some recommendations for other venues. And with that, I thank you for your attention. Please.
I think just your talk here today, and we'll all go home and talk, and so those kinds of things will grow just because of human beings talking. I mean, they're going to want to know more.
Yeah.
So.
Yeah, reach out. I'm always available. I do think, I mean, you can't avoid the news that basic science is in trouble right now, and We're scientists, we're problem solvers. So what's one way to solve a problem? Well, to get out and talk about what basic scientists do so that it's better understood. I mean, most people don't know that it takes 15 years to go from a basic science lab into clinical practice. That's a best case scenario, right there. And imagine, when I was working on these slides, I was thinking about, I mean, imagine being the person who reviewed Marion Diamond's grant 30 years ago, the title of which is Environmental Enrichment for Mice. Right? I mean,
Yeah.
that would not get funded in today's climate, right? You should be like, this is ridiculous, right? And so that's important too. I mean, we're learning. The basic scientists are also learning how to navigate. But I do think that we have a responsibility that we've neglected to spend more time in the community. Please.
Have you, well, I'm sure you're aware of the research on music and how people with dementia, because music uses so many different parts of the brain, that someone who's catatonic can actually start talking when they hear music.
Yeah. It's fascinating.
Yeah.
An individual who have mild to moderate strokes in the Broca's region, which is the part of our brain that helps us to form words, pull words from our vocabulary and then put them in grammatically correct sentences. If you have a stroke in that region of your brain, you can still sing. Right? And so part of this is the distribution of labor in our brains. And even though we parallel process a lot, which means that you have to lose a lot of neurons before you see a cognitive deficit. That's why Alzheimer's is so insidious, because there's that precipitous drop in neurons, but symptoms don't come until there's a lot of loss. Even though that's the case, we still specialize. And so the fact that music therapy can work in individuals where other therapy doesn't work is because it uses a different circuitry, an underutilized circuitry. So maybe
More areas of the brain, too.
more areas of the brain, right? Music stimulates a wide swath of brain, especially familiar music, because we have emotional connections to familiar music. Yeah. It's really interesting if you put a layman in MRI, functional MRI, and play the music, quite a lot of the brain lights up. But if you put a professional musician in an MRI and have them listen to music, different areas light up. And so, yeah, but part of it's because of the broad expression, representation of music. Thank you for the question. Please.
I've been part of a longitudinal study since 1957.
Wow.
How much interaction do you have with longitudinal study at the university?
Who's the PI? What?
I knew you were going to ask that.
So I've been here for 2 years, and I know many, but not all people. There's a very large neuroscience ecosystem on
Okay.
campus. So there are neuroscientists in my department, in the Department of Neuroscience, but there are also neuroscientists in neurosurgery, psychiatry, psychology, biomedical engineering, and neurology.
Okay.
Okay, so there's a really broad ecosystem that's both basic and clinical sciences. Can you tell me one key word about the longitudinal study?
Well, in October, I spent 3 days at the university going through the cognitive testing that they went through. And I was thrilled with it. I was really taken to it. And ironically, when you show your slides here, I can remember almost everything that went on during that 3-day process because it was so stimulating.
Because you're so stimulating.
Yeah.
And it's new. And you're
Yes.
Right? So thank you for sharing that example, because you know from your own experience that
Yes.
being in a novel environment is very stimulating. Yeah, I'd love to know more about the study that you're involved in, and if you want to email me the PI.
I will do that.
Is it Kalin? Kalin?
Sounds familiar.
Okay, that would be my guess. Male? Is it male?
Yeah.
Okay. I think it's, Ned Kalin has a very nice longitudinal study. Then there's also a guy named Sterling Johnson. He also has a very nice longitudinal study. So the research at the University of Wisconsin-Madison, as you know, right, is renowned.
Yes.
And so we're one of the top 10 in the US in terms of, well, here's the caveats. We're one of the top 10 in terms of research spending in state universities. Right? And so it's still good. And so it's a real powerhouse. So thank you. I'd love to hear from you.
Okay.
Please.
Do you have anything specific for the super agers, what they did to become super agers, or they're just anatomically super agers? Okay, and then
Go ahead.
and then anything else to slow down the onset of cognitive decline?
So there does not seem to be anything that is genetically unique about superagers. Okay? So that means that all of their superness comes from experience. Experience can change the way some genes are expressed, right? So it's a dialogue between experience and genetics, but there's no clear common sort of genetic signature in the superager. So you're not born a superager; you make yourself a superager.
Through nutrition, exercise, novelty, the enriched environment.
Exactly. And so those
I
are
know
the
they have the pockets in the world of superagers, and I've watched those videos. When in California was it, and when, I don't know, but.
Yeah, I've seen those too. I think that part of it, so this is gonna be hard to hear. I think they're all in climates where you can walk and be outside 365 days of the year. And so that's part of it too. If part of the culture is to, go for a walk on the beach every morning, that's easier to do in some warmer places than colder places. But I don't have any more specifics other than the classes the categories that I said.
Sure.
Challenge, enrichment, social interactions.
Same with the cognitive decline.
Streaming, same with cognitive decline. They're the same.
Yeah.
The neuron loss and the cognitive decline are the same. One is causing the other.
So I was just going to tag that to say experiences and all of that you've just said at an earlier age. Say you've got a 40-year-old that's just now going to start jogging on the weight room or whatever. How does that compare to someone who started this in college and or even high school in sports?
It's a really interesting question. We just don't have the data to do a rigorous statistical comparison. But I say, enrichment, let me answer this question first, and then I'll make another point. I argue that anywhere along this slope, right, this is really, I've been focusing on this synapse loss and this cognitive decline. And so any enrichment that's done in that timeline will prevent that cognitive loss and volume loss. And so there are some, I just can't speak to that. We just don't have, enrichment is relatively new. It's being applied particularly to just understanding what our brain likes. I mean, we were so wrong. We thought our brain liked routine. And for decades, we thought our brain liked routine. Our brain does not like routine, it likes the opposite. So that's sort of a new understanding. And it's being applied to primarily young children and older adults. So my mother's is 88 in assisted living in Algonquin. And it's amazing. They change the pictures in the hallway. Right? Every couple weeks, they change all the pictures in the hallway. And so, and that's how much this is This is how ingrained this is now. We understand this. The place where she lives understands the importance of the novelty, and they change all the pictures in the hallway, do a lot of other things. So this is just, you know, being embraced, but especially it's being embraced at the times where this slope is the highest. Early childhood and late adulthood.
One other question before I let you go. On that spectrum there, Alzheimer's, schiz, normal, and autism, is there any graphs or anything that one can find other than the book that's out, the grieving brain? You know, say one has been involved in grief at an early age. Life has kind of taken that toll in the whole spectrum.
Yeah.
But do you have any books that one, because there is a, you know, it's more, it's not fiction, but it's called The Grieving Brain, and I don't know the author of that, I don't remember.
Okay.
But I was just wondering if there's something we could access to see, in spite of everything you've said, educators on the grieving brain kind of comes in there with so much more information. Like, for instance, grief experts say, okay, in your grief, you have to have a habitual schedule in order to get through this tragic grief. So, okay, you're developing, I get up, I take off the alarm, I do my devotions, I go to a support group. But, you know, you look at all of those things that might be habitual. Would one say contradictory to grief getting through? Okay, maybe tonight I'll do my devotions at 10 o'clock and I don't do them at 8.
Yeah.
Or take my walk, you know. And then with that, when you do treadmill versus outdoor nature, it's like, I would love to know what that is. Because, you know, for 8 months or 9 or 10, we can do that outdoor walk, which I relate to. Have only done that horrible thing in the downstairs called treadmill, and I've already realized the differences in how I'm navigating everything in my life, including grief, with that change.
Yeah. So so thank you for sharing that. I'd be happy to look into references for you. So none come to mind, but that's only because it's outside my area of expertise. I think that the enrichment, all of these things that we're talking about, enrichment can bring any brain from its current state to the more enriched state. I believe that. But I think that there are also, so this is getting changed too. So there are also times immediately post-trauma or even immediately post-stroke, completely different example, where the practice has been for decades to let everything just sort of settle down, right? Get into routine and settle down and just and bring everything down to baseline. Right? So I can speak to stroke. For stroke, we know that the brain is the most adaptable immediately after the stroke, and most stroke patients are not getting physical therapy or occupational therapy right after the stroke, right? So we need to start that sooner. There would be more rehabilitative benefit if the therapy started sooner. It's not my call. There's a lot that me standing here, I'm a basic scientist, me standing here, you know, I can't make recommendations to the clinic, but that's what we know. I think that the old way of I don't want to say it's the old way of thinking, the standard way of thinking is to introduce routine after trauma until everything settles down. I don't know if that's ever been tested with a enrichment model, but I'd be happy to look into it for you, and I'd be happy to get back to you. Thank you for sharing.
Is there any research on how brain fog in COVID is it different than cognitive decline, or does it lead to cognitive decline? I know it's pretty new.
It's new, and so we don't have longitudinal data, but there's a real brain fog. There's a couple different brain fogs. One happens during COVID. One happens during chemotherapy. Also, there's a very well established brain fog. These are different. These are states that are interfering with memory formation, not a change in your capacity to make memories. So there's difference. So it's like the hardware's there, the hardware's fine, but the software is not making the connections.
But you don't have enough data to see whether that's a precursor to.
There's not enough data. There's not enough data. There's certainly not enough data. It's too soon with COVID.
Quick question, with alcohol related to cognitive decline, I know alcohol does not kill neurons, but it interrupts the synapses. Does it kill the synapses, or what does it do? Or is it just a temporary, or when it ends up being Alzheimer's induced, through alcohol, does it kill it, completely dissolve that synapse, or?
So alcohol is so complicated because it acts on multiple different targets in the brain, multiple different brain regions. There's idiosyncrasies, it changes, it impacts, so some people adapt and some people sensitize to alcohol. And so it's highly idiosyncratic and also complex in terms of all of the targets. That being said, the things that can be said is that information flow through the circuit. I mean, this is being done in humans, so we don't have a synaptic level. Information flow through circuits is really decelerated, right? So it's the slow thinking. And if activity is what strengthens the brain, reduced activity is going to not maintain it or result in weaknesses similar to this. That was a vague answer because I don't want to speak out of turn.
Sure.
We know a lot about addictions to chemicals that have a much more focused impact on neurons. Alcohol is really tough because of the idiosyncrasies. It does cause brain damage. Alcohol abuse can cause brain damage, and I assume and don't know. I'm sorry to say that is associated with neuronal death, long-term consequences.
Thank you.
Thanks so much. Nice to meet you.
Thank you so much.